All right, that looks great. So first up today, we have Meg McEachran. She is a PhD candidate in the Conservation Sciences program at the University of Minnesota, and I am her advisor. Meg is interested in many things, but as you'll hear in our talk today, she's very interested in applying interdisciplinary approaches to solve natural resource issues, in particular those that involve invasive species and wildlife disease. So take it away, Meg.
Awesome. Thank you, Nick. And thanks everyone for attending. I'm really excited to talk about this work with you. It's approximately 3 of my dissertation chapters, so there's going to be a lot of fast-paced information, but I hope that at the end you'll take advantage of the time for questions and we can have a little bit of discussion. I'm really curious to hear what many of you have to say. So with that, I'll be talking today about assessing the risk of fish pathogen introduction via the illegal release of live bait fish in Minnesota. In an era marked by the globalization and rapidly expanding trade in live animals, the spread of invasive species is a major threat to conservation, particularly in freshwater environments. A famous example of this is, of course, the red swamp crayfish, which is purposefully moved for the pet and food trade all over the world, but has become a problematic invasive species in many places where it's been established. In particular, invasive pathogens can be very difficult in baiter to deal with because they can pass undetected in otherwise relatively innocuous commodities and animals, and they can have population level impacts on native species of concern. One of the most famous examples of this is, of course, the chytrid fungus, which has spread globally via the trade of live amphibians and has resulted in the declines and some extinctions of several of those amphibian species. So against this global backdrop of trade and the potential for animal disease to be spread via these pathways, the release of live bait by recreational anglers has been identified as a particularly high-risk pathway for the spread of invasive species and fish disease, especially in the Great Lakes region and in Minnesota, where I'm located and where my work is focused. For those of you that are not really familiar with this system, small fish such as the fathead minnow shown here are a very popular live bait among recreational anglers. In Minnesota alone, it's a $2.4 million industry and supports an even larger multi-billion-dollar recreational fishing industry, which is of course also very culturally important in Minnesota. In Minnesota in particular, about 60% of the bait is sourced from the wild, harvested by commercial harvesters. And the remainder is coming from aquaculture, raised on farms here in the state. However, on the other side of things, pathogenic microbes and non-native species have been found in the bait supply in Minnesota and in other states. And in order to address some of these concerns, many places, including Minnesota, have made bait release, dumping the leftover bait fish into the water, illegal. But we know, as with most conservation regulations, compliance is not 100%. So combining this high volume and popularity of the live bait fish with the fact that there's minimal disease testing, low traceability of shipments, and lots of complexity and uncertainty regarding this pathway means we have one big mess when it comes to understanding the risk of live bait. So my project attempts to untangle this mess by asking, what's the actual risk of pathogen spread via live bait fish release, and what can we do about it? Taking a step back to understand the full pathway, we have to understand that a series of steps has to occur in order for a pathogen of interest to spread along the live bait fish pathway and affect fish of concern. First, the pathogen, of course, has to be present in the live bait fish, whether they're harvested from the wild or sourced from a farm. An angler has to purchase or harvest those bait fish, and then, of course, release them at the lake where the pathogens can interact with the natural environment and potentially impact fish species of concern. The first part of my talk will focus on this kind of first module here, which is to identify and prioritize the hazards that are out there. And the second half will focus more on quantifying the risky behaviors and understanding how this whole pathway interacts as a whole to result in the risk to wild fish. So our first step, as I said, is to identify and prioritize the hazards. Those of you that work in this field know better than I that there are hundreds of microbes, if not thousands, that are regularly found in aquatic environments, including in fish, but a very small percentage of those actually cause disease and are of concern to us. So we have to strike a balance between a precautionary approach that tests for and regulates all pathogens, and at the far end of that approach, the ban live bait, with the other side, which would say we allow everything and test for nothing, and only reactively manage outbreaks after they've occurred and caused significant damage. So to strike a balance between these kind of two extreme approaches, our group decided to pursue an evidence-based hazard prioritization approach. This seeks to balance the benefits of live bait use that I mentioned before with the risks that it could be spreading invasive species and fish disease. And the purpose of this was to create a risk ranking framework for pathogens in the live bait fish supply and apply it to the Minnesota bait industry to identify priority pathogens. In order to do this, we created a kind of filtering framework to focus our interests on pathogens that were particular to the live bait fish supply in Minnesota. So first, if the pathogen was listed or considered by any of the relevant authorities, including international, national, state, and regional authorities, it was included in our potential list. The second filter we used was whether live legal bait fish species could be natural hosts and able to transfer the pathogen. If bait species were not able to carry it, we're not interested in it for the purposes of this study. That's not to say that viruses and other pathogens can't be transmitted in earthworms or leeches or crayfish, but again, for the purposes of this study, we're focusing on live legal bait fish species. And the final filter we applied was whether fish of concern were susceptible. Now, fish of concern sounds a little bit subjective, and the truth is, it is a little bit subjective, but the idea was that we wanted to focus on pathogens that could affect sport fish or fish that are otherwise of concern for the Minnesota DNR, including threatened and endangered species. So if a pathogen made it through all of these filters, it was selected for consideration in our hazard ranking exercise. 15 pathogens made it all the way through these filters, and we then took these 15 pathogens and assessed them according to 7 risk ranking criteria that we developed. The 7 criteria were the current distribution in Minnesota, the colonization potential for Minnesota, ecological and economic impacts if established, the likelihood and prevalence of this occurring in the bait supply, and the host species that the pathogen could infect. We also elicited input from 25 experts, I think some of whom are in this room, so you'll probably be excited to see the results of your work here. We elicited opinions from 25 experts and asked them to weight the criteria according to the importance that they thought that they had, to allow for different criteria to have a greater influence on the risk ranking. And finally, these weighted criteria were combined using the following equation. And here's what we found. Our pathogen ranking framework identified Asian fish tapeworm, which is a generalist macroparasite, Ovipliostaphra ovariae, a microsporidian parasite of golden shiners, and of course the infamous viral hemorrhagic septicemia virus as the top 3 pathogens. In general, the hazard prioritization framework was able to distinguish between these pathogens, and it identified pathogens that had been identified as priorities in other frameworks as well. So our results agreed with the ways that other people had been assessing these pathogens. So we did that. We identified which hazards were out there and which were of the most concern using our framework. Check. Now we have to be in the business of understanding how the rest of the pathway works and how these diseases are allowed to proliferate along the pathway. And in order to do that, we need to quantify the behaviors that allow the transfer of these pathogens from the bait shop all the way to the lake where they may be released and cause problems for the wild fish there. I'm not going to talk a lot about this because there's a lot of behind the scenes data, but I wanted you to know how we got the data for the rest of this project. And again, the purpose is to quantify the bait-related and fishing behaviors of licensed anglers in Minnesota. So we administered a mailed paper questionnaire to 4,000 annual license holders across the state and around the country. We received a total of about 670 responses. Many Minnesota counties were represented, as well as several out-of-state respondents. So we had a pretty good response rate. The results of this I'm not going to show right away because I want to talk about them in combination with the rest of the project. So stay tuned just a moment. So once we quantify the behaviors of interest, we have to put it all together. We have to understand what's the number of fishing trips that results in the release of a fish infected with a high priority pathogen in a given fishing season. We've identified the hazards, we've quantified the risky behaviors. Now we have to figure out what's the probability that every single one of these things goes wrong. To think about it in terms of a conceptual framework, you can look at each of these steps in the pathway as an individual probability of occurring. And if any one of these steps doesn't happen, then there's no risk. For example, if an angler doesn't use live baitfish, then they can't transmit pathogens in their live baitfish. If an angler uses baitfish, purchases susceptible fish, purchases an infected fish, but doesn't release that fish, then it's not considered a risky trip. So we described each of the probabilities of these steps occurring using a probability distribution parameterized by the data from our angler survey, which I showed a couple of slides ago here. The single trip probability of riskiness is then multiplied by the number of trips per year per angler and the number of anglers to get the total number of risky trips that occur every year for a particular pathogen under a particular set of conditions. So I want to go through now and explain to you what we got for each of these steps. And we assessed these under 4 different scenarios. I only have 4 to show you today. There's lots of other things and lots of pathogens we're investigating, so stay tuned. But for today, we're going to talk about these things. First, we assumed a baseline condition, what's going on right now given the information that we have. Second, we wanted to assess the effect of reducing release behavior. What if we could cut the rate of release by 50%? What's the resultant effect on the number of risky trips? Third, we wanted to address whether an outbreak had a significant effect on the number of risky trips in the absence of any surveillance. And finally, we wanted to look at what would happen under an outbreak condition in the presence of targeted surveillance being implemented so that the pathogen could be detected if it was occurring. So first, the probability of using live baitfish. Unsurprisingly to me, baitfish are really popular in Minnesota, and about 70% of our respondents reported that they use live baitfish. When we talk about the probability of using susceptible fish, we have different susceptibilities for each of the species listed here and each of the pathogens, our VHS, OBO, and Asian fish tapeworm. But for today, I'm focusing just on our highest ranked pathogen, Asian fish tapeworm, to kind of illustrate some of the concepts here. But the idea is that not every species is susceptible to every pathogen, and the model accounts for that. The model also accounts for the number of fish being purchased. So the probability of an angler purchasing at least one infected fish is a function of both the prevalence of that pathogen as well as the number of susceptible fish that that angler purchases. So you can see between these different scenarios, what varies is the prevalence that we allow the pathogen to have in the model. And the mean number of susceptible fish purchased does not vary. So the anglers buy the same number of fish in all of these scenarios. The other thing that's allowed to vary is, as I mentioned, the probability of release. The current rate of release is here, and the reduced release is listed here. So based on our survey results, we found that about 20% or 1 in 5 anglers admitted to releasing their live bait in the water. So we allowed the rate of release in the model to be 20%. And this is a preliminary results slide, but I want to show you where we're headed and what we're thinking about in terms of these scenarios. So first, the baseline scenario for Asian fish tapeworm. If you recall, the baseline scenario, the prevalence was allowed to vary between 0 and 2%. The mean number of susceptible fish purchased was 26.2, and the probability of release was allowed to be 20%. I should say, let me clarify, these are mean values. In the actual model, they're stochastic, described by a probability distribution. So there's a range of values that these parameters can take on. For simplicity's sake, I'm demonstrating just with the mean values here. But when you run the full stochastic model, you get a number of outcomes. In the baseline scenario for Asian fish tapeworm, where the prevalence was allowed to vary between 0 and 2%, the mean percent risky trip probability was 0.77%. So what that means is only 0.77% of trips that occur are risky. However, you have to remember that Minnesota has over 600,000 licensed anglers, and some of them take a lot of trips per year. So that 0.77% translates to over 150,000 risky trips that could be occurring per year right now, with Asian fish tapeworm allowing the prevalence to be up to 2% in the bait fish that are susceptible to it. The next scenario we addressed was looking at whether the rate of release could have a significant effect on the number of risky trips. And as you can see, we did have a reduction of almost 50% in the number of risky trips. So we have 0.43% risky trip probability, which translates to about 75,000 risky trips a year. So we halved it. That's good. What if there was an outbreak of Asian fish tapeworm? Under the outbreak scenario, we allowed the prevalence to vary up to 20%. And this is in the absence of surveillance. So without any surveillance, without any detection or prevention happening for this pathogen, 3% of trips were considered risky. And when you multiply that over the millions of trips taken by anglers every year, that results in over 600,000 trips that could result in the release of an infected fish every year. And that's just a mean number. It's a little difficult to see because most of the trips are not risky, and that's what's being shown right here on this graph on the right. So as I said, I'm just showing you in these columns, these are just mean values, but because it's a stochastic model, we have a range of uncertainty and variability in that estimate of the number of trips. And that's illustrated in that graph on the right. So on the right side, the probability density is the y-axis, and along the x-axis is the number in millions of trips that were named risky by the model. So as you can see, most of the trips that occur are not risky. That's why we have this huge spike down here. But you have this distribution, this very long tail of trips occurring that are risky. The mean number was 630,000 risky trips. But as you can see, or maybe you can't see, it's such a small dot, way out here on the right. Under one of the 10,000 simulations that we did, almost 108 million, I think it was about 104 million trips occurred that resulted in the release of a fish infected with Asian fish tapeworm. So again, while most of the trips that are occurring are not risky, it is not beyond the realm of possibility. And the model says it's about a 1 in 10,000 chance that over 100 million risky trips are occurring every year. So putting it all together, you might say, so what? Approximately 1% of trips were risky out of over 16 million, but given the high number of anglers and the millions of trips that they take every year, many risky trips could potentially occur. And this increases with the prevalence of the pathogen and the rate of release by anglers. There's a number of assumptions and pitfalls and uncertainties associated with this type of approach, and I wanted to address them and acknowledge them because they definitely have an effect on the way we think about things. One of them is poor surveillance. Because baitfish are pretty poorly understood and their health status is often not very well known, we have unknown pathogen prevalence for a lot of the things we're working with. Asian fish tapeworm, as I mentioned, was our highest ranked pathogen, but it is not currently surveilled for in Minnesota. It has been found in other Great Lakes states, and that's why we allowed it to enter into our consideration. But right now in Minnesota, there is not widespread testing or surveillance for this pathogen. Another point about the model is that it's not geographically explicit. We did find that rates of release did not vary among geographic regions. It's not like some anglers are worse than others, but some areas are under more fishing pressure. So that results in a higher number of release events happening in those areas. In this current version of the model, that is not taken into account. We also characterize the risk across the whole year. So you get a yearly number of risky trips. And I don't currently have information on how that might change by season or by month. In addition, we make the assumption that this is a worst case scenario, that every introduction of a pathogen could result in the release of an infected fish and the infected fish being able to establish that pathogen in that environment. In truth, as we know, environmental factors, host species susceptibility, fitness of the pathogen, all of these things go into whether or not a pathogen actually will be established. But we're assuming a worst case scenario here in this model. However, overall, more introductions result in a higher probability of establishment. So we want to be in the business of reducing this probability. Secondly, our simulations shown here only included anglers with an annual license. We don't include anglers with more short-term licenses, such as daily or 3-day licenses. And so there's about another 600,000 anglers who are not included in this model that could be contributing to the risk of pathogen spread. And finally, there's a significant chance of non-response bias. Because bait release is illegal, it's possible that anglers are they feel the need to underreport their rate of release. So the actual rate of release is probably higher than the 20% that we got in our survey response. In the future, we have a lot of exciting things coming up. We'll complete the scenario analyses that I illustrated here today for all of our pathogens of interest, including VHS and OVO. And the next step is to try to understand how we can go about reducing that illegal release behavior, understanding why anglers release bait. And that's the next phase of the study, is to examine risk perceptions and motivations for illegal bait release across the Great Lakes states, and to take those insights and optimize messaging and enforcement strategies to reduce risky behaviors among different segments of anglers. If we could reduce these risky behaviors within the current regulatory framework and reduce the risk of fish pathogen spread via this pathway, we can continue to support the bait and fishing industries while also preventing and reducing the risk to the wild fish species that we all care about so much. With that, I'm right at 20 minutes, so I'm going to take questions. Thanks so much.
Thank you very much, Meg. Great talk. If you have any questions for Meg, feel free to unmute yourself, or you can put them in the chat box and I can read them to her.
There's tons of stuff I didn't talk about, so please ask questions. I hope I have an answer for it.
Hi, Meg. This is Tom Locke from Michigan State University. Excellent talk. Thank you.
Thank you.
So I guess I'm curious. One thing that I've always wondered about in terms of bait is the distinction between like released bait fish and then bait fish that are just simply used and say maybe consumed or fall off the hook or something. So I'm curious just basically on your thoughts about that.
Yeah, every time I give a talk like this, that question comes up. So I'm glad you brought it up. He's not a plant. The reason we focused on release is kind of twofold. One is you all are, fish disease experts and know better than I, but a live fish is going to be a lot better vector of disease than a dead or a half-eaten one. A live fish is going to allow, especially a virus, to continue replicating in it. It might be swimming around, interacting with other fish, potentially spreading it or shedding it in the water. So a live fish is just going to be a more competent vector in most cases than a dead or a sort of incidental contact between a fish on a hook and the water. The other reason we focused on bait release is it's illegal. It's a behavior in Minnesota that is currently regulated. And rather than try to, I'm going to use a punny phrase here, open the can of worms of discussing banning live bait in Minnesota, we decided to just start with the baseline understanding of how this regulation is working, whether or not people are complying with it, and if not, what can we do to get them to comply with current regulations before we start the discussion of whether or not we should consider banning live bait. I did have somebody at a conference come up to me, a very avid fisherman, and kind of point at me and say, when they ban live bait, I'll know who to blame. So there's definitely that perception that that's the direction this project is headed. I'm definitely not there yet. We're just trying to figure out what to do about illegal bait release.
Thank you.
Hey, Meg, we have a question in the chat box here.
Sure.
They're curious on how transferable your model might be to invasive species, or maybe more broadly, other types of hazards that move through pathways.
Yeah, so we actually designed the model such that it could be transferable. The only things that kind of would differ would be some of these things back here in the susceptible and infected fish. You'd of course have to have parameter values for your population of interest. So the survey we did is of people who fish in Minnesota. We don't have data on other states and other behaviors, but you could use a very similar framework to assess the risk of other invasive species. You'd have to have data to parameterize the human behavior, but also some of those, if prevalence is the word you want to use, maybe likelihood of occurrence of the particular invasive species. You have to have some information regarding how often that particular invasive species occurs in the bait supply and the likelihood that it gets scooped up and sold to an angler. One kind of insidious thing about pathogens is they could be present even if you do visual inspection for days. You might not catch a pathogen. So all of those things would have to be taken into consideration, but it's definitely doable within this framework. And I should also say within the framework of hazard prioritization, some of the things that have to be tweaked, including like host species doesn't make a lot of sense for an invasive plant or invertebrate, but that series of questions can be brought to bear for other species as well.
Great, we have time for one more question if anybody has one. Awesome. Well, thank you very much, Meg. Great talk.
Thank you. All right, I will say stop share.